Hello and welcome. In this video, we will be talking about chromatography. Chromatography plays a role in multiple aspects in our everyday life, such as creating vaccinations, food testing, drug testing, and forensic testing. In chromatography, components of a mixture are separated, identified, and purified. In this lab, we will use fast protein liquid chromatography, which purifies large molecules like DNA or proteins. There are a number of different types of methods used to carry out FPLC, all working on the same principle.
Size exclusion chromatography separates molecules based on size and shape. Here, larger molecules cannot enter the pores in the gel. They simply pass through and are eluted first. Smaller molecules, however, enter many pores in the gel and have more volume to travel through. Therefore, they travel slowly and are eluted later. Ion exchange chromatography separates molecules on the basis of charge. The column, sometimes referred to as the ion exchanger, can be positively or negatively charged. Separation depends on what pH the protein is stable at and what protein charge is at that pH. The protein that does not bind to the column will elute immediately, whereas the protein that binds will not elute until the elution buffer is added. Lastly, affinity chromatography separates based on the interaction between a specific ligand and the protein of interest. During this process, keep in mind that the ligand must be bound to an insoluble matrix in the column, and the protein must reversibly bind to the ligand, or else we will not be able to get the purified protein.
In protein purification, fusion proteins are usually used to detect the protein of interest. A fusion protein consists of the protein of interest and a tag like glutathione S-transferase, short for GST. GST-tagged proteins are usually purified using affinity chromatography where the ligand is glutathione. The first step of GST-tagged protein purification is to add a solution that equilibrates the column. This ensures that the proper buffers are present for binding and the solution is at optimal pH. Then, the sample of protein mixtures must be loaded. The undesired proteins that do not bind pass through the column and elute first, whereas the desired protein binds to the glutathione ligand that is bound to the insoluble matrix. Next, the column must be washed to remove any undesired unbounded molecules to prevent contamination. Finally, the elution buffer is added, which contains excess reduced glutathione to remove and purify the GST-tagged proteins. The desired proteins will elute and can now be collected.